Hello everyone. Today I'm going to be talking to you about light-induced synthetic organelles and how they can be used to increase the metabolic flux of a desired product within enzymatic pathways. The process of enzymatic metabolism from initial substrate to final product is complicated and typically requires multiple steps to get there. Several enzymes can use the same substrate to make a variety of products without much discrimination between them. But what if we want only one specific product and don't really care about the other ones? How can we sway the enzymatic process to preferentially create only one final product? This is the key question researchers wish to find a solution to, and they came up with a brilliant one: the use of optogenetics to synthetically create organelles. In order to take a substrate and have it reach a desired product through enzymatic metabolism, it must follow a very specific pathway of enzyme interactions. To increase the chance that these interactions occur, the enzymes of interest would need to be closely co-localized so that the product of one enzymatic interaction quickly moves to the next enzyme of choice and not a different one. This is the motivation behind using synthetic organelles. So what is a synthetic organelle? A synthetic organelle is the collection of proteins within a cell that are of a separate phase so that the proteins can serve a specific purpose. You might think of the Golgi apparatus or the endoplasmic reticulum, for instance, as a natural organelle. A synthetic organelle is created by combining certain proteins together to form their own semi-structure. We refer to this structure as either a droplet or a cluster, depending on how close together the proteins are. A protein called FUS, or fused in sarcoma, is useful in creating phase separation within a cell. FUS utilizes pi-cation intermolecular interactions to associate together. Adding this protein onto enzymes would allow them to combine and phase separate. But the problem is now, how do you force these enzymes to associate together on command? This is where optogenetics come in handy. Optogenetics is the process of using light to manipulate a chemical environment in some way. Certain molecules are reactive to light, which can change their conformation, and in turn their function. In order for the desired enzymes to co-localize, an optogenetically active protein sequence called Cry2 can be introduced onto enzymes. Cry2 is referred to as a staple protein, meaning that under the influence of light, it closely associates with other Cry2 proteins in the area. So now, we've created synthetic organelles by grouping certain proteins together to form a phase-separated droplet within a cell, using FUS. That droplet can now be actively created and dissociated on command using Cry2. The droplets co-localize enzymes, increasing the production of our desired product. The alteration of natural pathways using synthetic organelles provides a possible alternative to industrial chemical production, and could one day be the main source for biofuels and plastics. If you want to learn more about how the researchers created the synthetic organelles and what chemical pathway they altered, you can follow the link on screen and in the description. Thank you for watching.